Hello everyone, my name is Nicholas Picarella and the current events article that I chose to present was Microbially Enhanced Vanadium Mining and Bioremediation under Micro and Mars Gravity on the International Space Station. Okay, so in terms of who, when, and where, this study was conducted by the European Space Agency, or ESA, and it was dubbed the BioRock experiment. All of these experiments began on July 30th of 2019, and this article was subsequently published on April 1st of 2021. So in terms of where, this experiment had two components. One was a space experiment, and this took place on the International Space Station, or the ISS, and there was a ground experiment component which was performed at a NASA research center in Los Angeles, California. So in terms of some background information, we know that microorganisms are used in a variety of industrial processes. So this can include biomining, which is extracting metals from rock, or bioremediation, which is used to break down environmental pollutants. In terms of the biomining process, this can be applicable to other planetary bodies. So what this essentially means is that microgravity affects metabolic processes in microorganisms, and there have been studies that have shown that microorganisms are capable of being metabolically active in altered gravitational environments. And lastly, vanadium is a metal that is important in numerous industrial processes. So one such case is that vanadium can be used as an alloy to strengthen structural materials. And one cool thing that this paper noted is that vanadium can be used in the construction of extraterrestrial rovers. So now I'll briefly just explain what the purpose of this study was. So as the paper mentioned, the purpose of this study is to test the hypothesis that the gravity level influences the efficacy in which biomining could be achieved from basalt, which is an abundant material on the Moon and Mars. They do this by quantifying bioleaching by 3 different microorganisms under microgravity, simulated Mars and simulated Earth gravitational conditions. So as I mentioned on the previous slide, 3 different microorganisms are being used in this study to quantify bioleaching capabilities on altered gravitational environments. So what are the microorganisms that are being studied? The first is Sphingomonas discesibilis. This is a gram-negative, non-spore-forming, and non-motile bacterium. And previous studies have shown different bioleaching abilities in this bacterium. And something cool about this bacterium is that it was isolated from the soil crust in the Colorado Plateau. The second organism being studied is Bacillus subtilis. This is a gram-positive, motile, spore and biofilm forming bacterium, and something cool that they noted about this bacterium is that it is commonly used, and it has been previously used in a variety of other space experiments. And lastly, the third microorganism that is being studied is Cupridivrius metallodurans. This is a gram-negative, motile, and non-spore forming bacterium, and it has been isolated from metal contaminated and rock environments. And one more thing to note, this image on the right is just it is an image of a biofilm of Sphingomonas discesibilis. So briefly, just in terms of the methods of this experiment, basalt rock acted as the substrate for bioleaching, in which vanadium was hopefully extracted by the microbes. Each basalt sample was dry heat sterilized prior to the flight and prior to the ground experiment. So this experiment was broken up into 2 components, one being the flight experimental setup in which 36 samples were mounted and sent up to the International Space Station, and the second being the ground experimental setup in which 12 samples were mounted and experimented on in the NASA Center in Los Angeles. So for both experimental conditions, altered gravity conditions were simulated, so the first being Earth's gravitational condition, Mars gravitational condition, and Moon gravitational condition. So to reiterate, this study investigated the use of microorganisms to extract vanadium present in basalt rocks. These basalt rocks are commonly found on both the Moon and Mars. The 3 experimental conditions that they studied were microgravity, Mars simulated gravity, and Earth simulated gravity. In terms of their conclusions, the researchers found that S. discesibilis and B. subtilis actually can biomine quite well under altered gravitational conditions. However, the researchers also found that C metallodurans really did not enhance the leaching of vanadium in the three different experimental conditions. So overall, to conclude, these results demonstrate the possibility of microbial bioremediation and elemental recycling in space and on other planets. So why is this study significant? First, this study demonstrates that microbial cellular metabolism is in fact maintained in altered gravitational conditions. So based on these findings, we know that microbial growth and metabolic processes are in fact possible on other planets in space. The second reason why this study is very significant is that this study demonstrates that extraterrestrial biomining is possible on other planetary bodies. So therefore, in terms of colonizing other planets, we can efficiently biomine important metals, specifically for creating alloys used in construction. So biomining can also enhance the efficiency of mining capabilities, thus resulting in less sediment being destroyed on other planets. And lastly, this study is significant because it demonstrates that industrial biomining can aid in the construction of high-quality metal alloys in space. And these metal alloys can be used in nuclear reactor components, batteries, catalysts, and superconductors. So overall, this study may one day aid in human colonization of other planets within our solar system. And lastly, here's just the paper that I referenced for this presentation. It was definitely an interesting read, and it wasn't too difficult to read either. So if you're interested, check it out. That's all I have. So thank you all for your time. I hope you have a great weekend, and I'll see you in class on Monday.